Hey, yesterday we talked some biochemistry, boy. This is it. All right. So understand that biochemistry, the first time you see it, get comfortable that the first time it goes by, it's like a bucket of water in the face. You get it, you whack, wow, what was that? Right. And then you get it again and again and again. All right. And then after you see it multiple times, then you start to see the patterns, you start to see the flow of the information. Be patient with yourself, okay? Even if you're not a STEM major in college, you're going to get into classes at some point where you don't get the content the first time. Don't let that rattle you, okay? If you don't get it the first time, say, all right, I'm gonna get it the next time, or I'll get it the next time, or I'll get it the next time. That's called studying, all right? One of the interesting characteristics of whatever you want to label the students, high functioning, honors, advanced, IB, top 10%, whatever gate, whatever stamp you want to put on their forehead, those students actually tend to have pretty poor study skills on average because they can process content quickly. And so what sometimes happens, sometimes during IB, sometimes it's at the university. Some of you may not hit it in your bachelor's and you won't hit it till your master's, but you will hit a class where you don't get the content immediately. And sometimes that makes students panic. Don't panic. Just go, hey, this is what everybody else calls studying. And just take your time, find a place where you're comfortable and study the content. Whether you're a flashcard person, whether you write it out, draw it out, discuss it, whatever is your learning style. Cool. All right, Calvin cycle. What's the whole point? We have 3 items going in and we have 1 item coming out. Okay, what goes in? Carbon dioxide, ATP, and high energy electrons. 3 things going in: carbon dioxide, ATP, and high energy electrons. 1 thing coming out, G3P. Okay, now since it's G3P, as in glyceraldehyde 3-phosphate, right, there are 3 carbons. So since there's 3 carbons coming out, even though Campbell only shows 1, really 3 of those carbons go in. So it's really 3 carbon dioxide go in for every 1 G3P going out. Okay, now if Campbell's really wanted to make this accurate, there'd be 3 RUBPs here, there'd be 3 CO2s here, there'd be 3 intermediates here. But you start doing that and this image starts to get cluttered. So then students go, well, that's a cluttered image. All right, but then if you pull the 2nd and 3rd molecule out and just show the minimum, then students go, well, that's not showing all 3 of them. So really the author loses either way. But step back and realize what's going in and what's coming out. Carbon dioxide, ATP, and high energy electrons in with G3P coming out. Em, ask a favor. Could you take a look at that for me? Okay. Then this is a cycle. It's a circle. That's why it's the Calvin cycle. So you have RUBP here. Thank you. And there is 15 carbons here. So this whole left-hand side of the clock is 15 carbons. You have 3 coming in the top. So it's 15 plus 3, which means this other half, this over here, this is all 18 carbons. So it's 15 plus 3 gives you 18. Okay. Well, if 3 come in to make 18, then 3 have to come out. 3 carbons exit or are synthesized, produced, and then you go from 18 back to 15. All right, so be sure to step back and see the big picture here. On the left-hand side, 15. On the top, 3 coming in, and on the right-hand side, 18, and on the bottom, 3 coming out. Thank you. Okay, so let's put some names here. This, the fancy name, ribulose 1,5-bisphosphate. Okay, so this is ribulose 1,5-bisphosphate. No one calls it ribulose 1,5-bisphosphate. Okay, we all call it RUBP. So then you have RUBP. The capitalization there is important and the lowercase is important, right? Now, this is a 5-carbon molecule, okay? So you have, let's see, 5-carbon molecule. You have 3 of them. Therefore, you have a total of 15 carbons. You have 3 5-carbon molecules, right? Since you have 3 5-carbon molecules, 3 times 5 is 15. Then there is an enzyme here that is going to bond carbon dioxide, right? So this is CO2. It bonds carbon dioxide to RUBP. Any enzyme that bonds CO2 to a substrate is called a carboxylase. So this is a carboxylase and does carboxylation. Okay. Well, you read enzyme names backwards. So ACE is an enzyme, carboxyl group. So this is an enzyme that adds a carboxyl group to some substrate. Which substrate?
RUBP.
Yep. So then the fancy name, or the I shouldn't say the enzyme name is RUBP carboxylase. Fine. Okay. But we don't call it that, and I don't know why. We call this enzyme Rubisco. And I've never looked up the history as to why. I just know that it's called Rubisco. And I don't know how we got from this to this, because I think this enzyme name actually makes a lot of sense, but we call it Rubisco. Fine. So then you have your carbon dioxides, but there are 3 of them. So since there are 3 carbon dioxides, you are bringing in a total of 3 carbons. This is why 15 plus 3 is going to give us 18 carbons all over here. Now, when this carbon dioxide is first added to this RUBP, the carbon dioxide is added to the number 2 carbon. However, this is unstable, right? So this technically is only called an unstable intermediate. It is so unstable and it's so temporary, we don't even give it a formal name. It's just an unstable intermediate, okay? And what happens then is the bond between that carbon and that carbon, that bond right there, breaks. So when you have a 6-carbon molecule and the bond in the middle breaks, you end up with 2 3-carbon compounds, okay? So you originally have 3 of these unstable intermediates. Each intermediate has 1, 2, 3, 4, 5, 6 carbons. So if you have 3 molecules of 6 carbons, you have a total of 18 carbons. All right, so this unstable intermediate breaks in half and makes what's called phosphoglycerate. This 3-carbon compound is phosphoglycerate. Now, understand there's oxygen and hydrogens on the side of these carbons that we are completely ignoring. All right, we're ignoring a lot of chemistry here. We're simply focused on that carbon backbone. So this is phosphoglycerate. However, the phosphate is on the number 3 carbon, therefore it is 3-phosphoglycerate. However, people don't call it 3-phosphoglycerate. They simply call it PGA. Okay, so RUBP is bonded to CO2 to make PGA. Really how this works. RUBP is bonded to CO2 to make PGA. Okay, now we need some power to do the work that's going to add the high energy electrons. So ATP comes in. Uh-oh, my lamp is fading. Can you see it pulsing? Come on, baby. One more day. But what leaves is only ADP. Notice how there's no inorganic phosphate here. It's just ADP. Really? I might have to replace it in between classes. All right, so that's why there's 1 phosphate here, but there's 2 phosphates here, right? So this ATP drops a phosphate off. So now this is also a phosphoglycerate, but it has 2 phosphates. So it is a biphosphoglycerate, right? And since the phosphates are on the 1 and 3 carbons, therefore it is 1, 3 biphosphoglycerate. Okay, but again, nobody calls it that. We call it BPG. And this is where it makes me giggle when science people use shorthand, because you will hear people say PGA bonds to ATP to grab a PO4 to make BPG. And if people are familiar with the cycle, we go, uh-huh. And non-science people are like, what? Okay, it's just this substrate bonds to a phosphate from ATP. You now have 2 phosphates on your substrate, and you form a new product called BPG. Fine. Now it's time for the high energy electrons to show up from photosystem 1. Those high energy electrons are carried by NADPH. It's the electron carrier in photosynthesis. So here's those high energy electrons that were excited in the complex of proteins that are photosystem 1, almost said 2. So what leaves then is just NADP plus the proton, and notice how the phosphate leaves, okay? So since the NADPH is giving up its electrons, the NADPH is oxidized. What is dropped off are the high energy electrons. The BPG gains those electrons, therefore the BPG is reduced. The BPG is then reduced into glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. But once again, of course, no one calls it glyceraldehyde 3-phosphate. We all call it G3P. Now, notice how I haven't talked about the carbons at all. That's intentional, because have we added or taken away any carbons on this right-hand side of the cycle?
No.
None. So we have 3 PGAs. Oh, nuts. I wrote my parentheses the wrong way. Boy, how do you do that? Let's try that again. You have 3 PGAs for a total of 18 carbons. You have 3 BPGs for a total of 18 carbons. You have 3, where should I write this? Can I squeeze? No, I'm not going to squeeze them in. You have 3 G3Ps for a total of 18 carbons. So there's no carbon leaving here at all. There's no carbon entering here at all. It's all 18 carbons, okay? However, 1 G3P leaves. Every time the Calvin cycle turns, you produce 1 G3P. That leaves the process. So 1 carbon leaves. That leaves with 3 carbons, meaning that instead of having do you want to know the good news or the bad news?
Bad news.
Bad news. I made a mistake. Is that number right there correct?
No.
No, I'm too busy talking. What should that number be?
6.
Thank you. Sorry. There we go. I wondered why when I got down here, the little voice was like, this isn't right. I'm like, no, it looks good. I'm like, no, it's not. All right, so you had 6 G3Ps. 1 G3P leaves, which means how many G3Ps are here?
5.
5. And could you grab the lamp from the front room here? Sorry for the interruption again. 15 carbons. Okay. Now, there is literally a shuffling of carbons from here to here and from here to here that you don't have to worry about at all. When you're a junior in college, you'll take biochemistry and they will get into the chemistry here. The animations that I show you will literally just take the carbons and go like this. That's perfect. Thank you. So the name of this is triose phosphate. We simply call it TP. Some more ATP comes in. ADP leaves. And the TP is then converted back into 3 molecules of RUBP. Just like that. So, do you think students get overwhelmed with all of these details?
Yeah.
Absolutely, totally, and completely. Okay? Because you hand someone, thank you, a diagram that looks like that, and you go, "You've got to be kidding me." Okay? So, here's what you do. You see the big picture idea. Yep. Where do you want to start? Let's start at the very top, the very bottom, in the middle, on the left, on the right. Where do you want to start? I have no preference.
At the top.
At the top. What are we putting at the top? Yes. We're adding 3 carbons to the top. Okay. Once you add the 3 carbons, what then goes over here?
The unstable intermediate.
Totally. Unstable intermediate. Unstable intermediate then goes into?
6 PGAs.
Yep. PGA, then?
BPG.
BPG, then?
E3P.
E3P. Okay. Then what comes in over here? 2 things. Okay. This would be a phosphate from ATP and the high energy electrons carried by NADPH. All right. Then what comes out the bottom?
G3P.
1G3P. So you're then minus 3 carbons. That leaves you with G3P. G3P is then converted to TP. TP is then converted back into RUBP. Okay, 3 in, 3 out, 3 in, 3 out. That's how it works. Okay. How many carbons do you start with over here? Big marker, there it is. 15 carbons here. You add 3, how many are all over here?
18.
18. That's Calvin. Okay. Be careful when you see all of this detail. Get comfortable here first. Okay. RUBP, 3 carbons in, unstable, PGA, BPG, G3P. G3P, why is it special? Why is G3P special? Because it contains high energy electrons. This is what all macromolecules are made from in plants. This is a really, really, really special molecule. Really special. Okay. And then when we talk about cellular respiration, we'll talk about a process called glycolysis. And I will mention, we won't get into the detail, but one of the first things that cells do when they get macromolecules, they almost immediately start cutting it down into, ready? G3P. G3P you will find everywhere in metabolism. Okay, so 15 plus 3, 18, 18 minus 3 back to 15, RUBP, and then the steps. Any questions on this before we leave it?
No.
All right. Okay. Wow. All right. So then what you want to do, I need a ruler. So we're going to do, let's see, let's try to find the middle of this. Right about, right about there. So if we take a line up here, this, I'm going to take a line about like that, and then we take a line. There are 3 major phases to Calvin cycle. The first phase up here is carbon fixation. Fixing carbon is when you attach carbon dioxide to any substrate. So this is carbon fixation. Of course, RUBP, you attach the CO2, unstable intermediate. This is now called reduction. Phase 2 is reduction. What gets reduced in the Calvin cycle?
BPG.
BPG is reduced in the Calvin cycle into G3P. And then what's down here? Just regeneration. Regeneration of what's called the CO2 acceptor, which is basically RUBP, like such. Okay. Now, if you have the opportunity and you have access to color, it is helpful to take this and shade this whole thing in. So what'd you do in class today? Oh, I watched Mr. Manro color. You too can go to college and get paid to color, like such. Oh, that's right. We give Emma a hard time since she's going to teach elementary school, that all you teach in elementary school is colors. So crayons, the alphabet, crayons, letters, and and shapes. Right? That's so insulting. It's like when people say, "Oh, so in high school," they say, "So you just have to just tell the teenagers what to do." Like, oh yeah, that's all there is to it. You just look at the teenagers and say, "Do this," and of course the teenagers go, "Oh, I'm so motivated when you just tell me what to do." No, there's a little bit more to it than that. Like such. All right, and then I don't know. Do you want orange or red?
Red.
Red. You got it. Interesting. With the camera, it looks more orange. I swear. Here I give them a choice, and then it looks like I ignored their input. That's really funny. I highly value your input. Look at that. There you go. Beautiful. 3 phases. Fix the carbon, reduce your substrate, reform your acceptor. 3 phases. And this little baby right here, G3P, is what we call a metabolic precursor. G3P is a metabolic precursor for all macromolecules. From G3P, you can make lipids, carbs, amino acids, and nucleotides. All of them. Okay. So here's what I'm going to do. I need to post some animations for you, which should be. Let's show you this one first. How are we doing on time? 10:50? Oh, plenty of time. All right. So what do you think this gray thing is here?
The thylakoid.
That's the thylakoid. Okay. This is the thylakoid. This would be photosystem 2. This would be photosystem 1. And this would be ATP synthase. And you go, but Manro, we drew ATP synthase down here. It doesn't matter the location, okay? You need to know that photosystem 2, the electron transport chain, photosystem 1, and ATP synthase are all embedded in the thylakoid membrane, okay? So here's your light going into the photosystems, bouncing around in the chlorophyll. There's your electron flow. And then they're going to then show you how the protons are then pumped. They don't flow, the protons are pumped. They're forced into the thylakoid space by the proton pump, that cytochrome C in the middle. And here's your electron acceptor, NADP, forming NADPH. Then they show you the water. how the water is first broken by photolysis, then the water is oxidized. Let's show that to you again. Boom. So notice how the hydrogen in the water is broken into electrons and protons. So photolysis is simply the splitting of the water to oxygen and hydrogen. Then the hydrogen is broken down even farther into the electrons and the protons, true oxidation. Okay, the protons linger here in the interthylakoid space. The electrons go to photosystem 2 to be excited. And then once the electrons are excited, they flow across the electron transport chain and allow the pumping of the protons. Okay, you then have a proton gradient where these protons are going to flow here. Okay, the protons then flow from an area of high concentration to an area of low concentration by a process called chemiosmosis. Chemiosmosis occurs across ATP synthase. ATP is synthesized. I'll show you how in just 1 second. Okay, so this is a very simple animation that shows you that water goes in, sunlight, these are the light-dependent reactions, and they form NADPH and ATP. Cool. And YouTube is absolutely loaded, and I mean loaded with animation. So jump over to YouTube. I intentionally am using very fundamental animations here because I want you to go to YouTube and I want you to type in light-dependent reactions, photosynthesis, type in animation, and oh man, you'll find some really cool stuff. Could open that file, that'd be great. All right, so Calvin cycle, right? So here are your products of the light-dependent reactions, right? NADPH and ATP. They will then show you those big gray spheres for the carbon. Okay, there's your carbon dioxide with your oxygen in red. Now, what makes this animation cool is it shows you all three. Okay, so here's your three RUBPs, and you go, hold it. It's in the top right. It's a cycle. Be comfortable with the concepts, not the specific location, because they will rotate it on you. So RUBP. RUBP then is bonded to CO2. There's some chemistry magic. And you then get your PGA. You go, what about the unstable intermediate? This animation skipped it. Fine. Here are your 6 PGAs, right? But they're missing a phosphate on the end. These are only PGA, not BPG. So now the ATP is used to phosphorylate each substrate and convert PGA into BPG. That's why you made, or that's why the plant makes the ATP in the first place, at least inside the chloroplast. Here's your substrate level phosphorylation. And now you have BPG. Okay, now it's time for these high energy electrons to reduce these 6 substrates, right? NADPH is oxidized, the substrate is reduced, and you now produce 6 G3P. 1 G3P leaves, leaving you with 5 G3P, right? 6 minus 1 is 5. Perfect. Okay. Now that leaves you 5 G3P. There is some reorganization of the carbon backbone. This requires ATP to power the enzymes, and you are back to your 3 RUBP to start the process over. So each cycle, each turn of the Calvin cycle produces 1 G3P. So they're going to show you the second one a little bit faster. Okay, they will bring in the carbon dioxide. Then they will bring in the ATP. So here's carboxylation, right? RUBP carboxylase. Then you have substrate level phosphorylation. Now you get reduction of your substrate. You produce 1 G3P, and then your 5 G3P are reorganized. And then watch what happens over here with these 2 G3P. It goes into a glucose. Okay? It takes 2 G3Ps to make 1 glucose. That's why it's C6H12O6. So what we will do on Tuesday is I will show you a much stronger, more in-depth three-dimensional animation of this. I will also post it in Facebook. We'll then break out into groups. We'll present this content in a week, not next week, but the week after that. And you will be finding animations on YouTube and then showing the classmates and explaining them. That's when you really start to learn photosynthesis, when you have to explain it to. So I will see you this afternoon about 12:45, 12:50 in the Zoom link, and then we'll talk to some individuals from AstraZeneca. Should be super awesome. Don't forget to type attend, and I'll see you all in a couple hours.